Okay, good evening students. Today I am going to discuss about the Mendel's law. The Mendel proposed mainly three laws. What are they? Law of dominance, law of segregation, and law of independent assortment. Okay, today I will start the Mendel's first law. See what is the Mendel first law? Mendel's first law. See what tells about the Mendel's first law. See, this law tells about the every character is controlled by a pair of factors. See, every character is controlled by a pair of factors. See, what is the meaning of factors? So, factors means genes, remember. So, in this first law tells about every character is controlled by the pair of factors. That means genes. Always genes are formed as a pairs. Remember. Okay. Coming to the next point. In case any one factor is dissimilar, that means if any dissimilar factors are present, one factor dominate than other. In case of dissimilar factors are present, then one factor dominate to other. Remember. So this is the information about the Mendel's first law. So in this Mendel's first law tells about two major points. What are they? Every character is controlled by the pair of factors or genes, and in case of any one factor the factors are dissimilar, so one factor is dominate than the other factor. So in this Mendel's first law, like this information already we will discuss tall plant and dwarf plant. So if you take tall and dwarf, so when we conduct the crossing between the tall and dwarf, then what kind of plants we are seeing in F1 generation? Both are tall plants. In F1 generation both are tall plants. Then again we will conduct the self pollination between these F1 generation plants, then F2 generation plants. It forms the tall and dwarf. So this is about the law of dominance. Okay, exception to Mendel's first law, some other experiments are also conducted by the Mendel. So what is that means? The another type of dominance, that is called incomplete dominance, and also second one is called codominance. First I am going to explain about the incomplete dominance. In some of the plants we are seen in the incomplete dominance. See what is incomplete dominance? See, incomplete dominance. Incomplete dominance. So this incomplete dominance we are seen in the one specialized plant. The plant name is called snapdragon. Snapdragon. Remember? So in this plant we will see the incomplete dominance. See how to find out the incomplete dominance means. See, I will give one example here. Example 1. So I will take here two types of flowers. So what kind of flowers means? First I will take the red color flower. See, for suppose this is red color flower. See which type of flower it is? Red color flower. Then again I will select the another type of flower, that is called white color flower. For suppose this is white color flower. See, this is red color flower and white color flower. So I will take two types of plants. One plant produce the red color flowers and another plant produce the white color flower. Okay, in this red color means it consists of, so these character of the flower we are going to use some letters. For example, capital R and capital R. See, capital R and capital R indicates red color. So what about white color? White color means we will use the small r and small r. See, always what kind of letters we are using in the dominant character, the same letters we will write in small letters in the recessive. So in these two flowers, which is the dominant? Red color flower is the dominant. White color flower is the recessive. How to find out this? It indicates that we will use the capital letters. Always red dominant and always white is a recessive. See, this is called a red color flower. Red color flower. And this is called white color flower. This is white color flower. Remember? So the Mendel crossing between these two plants, white color producing flower plants and also red color producing flowering plants. So the Mendel conduct the crossing between these two plants. So in what kind of plants we are seen in F1 generation? See here, commonly what kind of gametes are produced from the red color flower. So these are the genes. They produce the two gametes here, capital R one gamete, here also capital R one gamete. Like this, here also the white color smaller, it also produce two gametes. What are they? Small r one gamete and small r one gamete. Then the gametes are fused to each other. See, this gamete is fused with this. So what kind of plant will produce here? Capital or small r. This is one plant. And here also it is fused with this gamete. Then it forms the capital or small r. See, this is one plant and this is another plant. Okay, in this process, what kind of flowers we are seen in the F1 generation? This is called F1 generation. So F1 generation means what? First filial generation. See, actually, law of dominance, Mendel's first law of dominance, capital R, small r indicates compulsory it is a red color flower. Why? R is dominant. Here also R is dominant, but it is not exactly similar to the Mendel's first law. Remember that. So the incomplete dominance is a exception to the Mendel's first law. Remember. So incomplete dominance is a exception to Mendel's first law. Remember, very important. So when we observe the incomplete dominance in the snapdragon plant, it is exception to Mendel's first law. Here somewhat difference we are notice. See what kind of difference we are notice, I will explain. So we will take the chromosomes. What kind of chromosomes are present? See, these are the chromosomes. Inside the chromosome consists of a double-stranded DNA like this. This is the double-stranded DNA. Here also double-stranded DNA. So this is belongs to capital R. Capital R means what? It indicates the red color. I will use a red color pen. See, this chromosome consists of capital R. Which type of gene? See, the gene which is present, a part of DNA we are called as a gene. So which kind of gene it is means it indicates the capital R. In the second chromosome, in the same location, here one more gene also present, and this gene, it is a small r. It is a small r. So what kind of changes we are noticed in the incomplete dominance means, listen carefully. Here the red color gene, it forms a normal DNA. So this red color gene produced from the normal DNA. So this is which type of DNA? Normal DNA. It is a normal DNA. Remember, it is a normal DNA. So in this normal DNA, it produces the normal gene here. Gene. Which type of gene it is? Normal gene. Then this normal gene produce the normal RNA. This is called normal RNA. Then this normal RNA, it produce the normal protein. Normal protein. Remember. So this normal protein is responsible to giving the red color. Which color? Red color. Understood? So when we observe the second type of chromosome, here some of the changes we are notice. What are the changes means? See here, a part of DNA we are called as a gene. Here also it is a gene and this is called DNA. So which kind of DNA it is means it is not a normal DNA. Why it is not normal DNA means some of the UV rays, ultraviolet rays will fallen on this chromosome, will affect on the chromosome. Then what will happen? The DNA, it converts into defective DNA. So which type of DNA here? A defective DNA. Defective DNA. Why it is defective DNA? Due to the effect of the ultraviolet rays. Not only ultraviolet rays, some environmental changes or some carcinogenic agents. Due to various reasons, in this DNA is modified into defective DNA. Okay, then the defective DNA, it produce the which type of gene here? A defective gene. Which type of gene it is? A defective gene. Then the defective gene, it produce the defective RNA. It also defective RNA. Then the defective RNA, it produce the defective protein. Defective protein. Okay, you have to remember one thing. Always the small r indicate recessive, and keep one thing in your mind, the small r is a defective allele. So it is a recessive. It is not able to giving the proper color to the flower in this experiment. Okay, this defective protein is a useless. It is not responsible for giving any color. For that reason, it gives which color? White color. So it is not giving any kind of color. So it is responsible for white color. And this white color is not expressed. So it is a recessive character in this chromosome. Okay, but here you are going to remember one thing. So if the DNA produce the gene produce the RNA, then RNA produce the protein. So in this case, here some changes we are notice. What are the changes means? If the formation of red color needs for suppose 100 grams of protein, but this gene or this RNA, it produce only 50% of protein only. So the gene produce the 50% of protein. That means it produce the less protein, remember. So there is no 100% of protein from this normal gene. For that reason, Here only 50% of protein only released. So this 50% of protein is not possible to giving the red color. Is not possible to giving a red color. For that reason, here this RNA or this protein, it gives the intermediate color. What is the intermediate color? Means it produces the pink color. Which color? Pink color. So in this F1 generation, what kind of flower we are seeing? Means pink color flowers. So it is a recessive character. It is a recessive character. Okay. So why here pink color flowers we are seen in the F1 generation? This is called F1 generation. So we will take the so here see, these are the F1 plants. So which kind of flowers these plants produce? Means pink color flowers. Both are pink color flowers. So then again we are conducting the self pollination between these two F1 plants. See, already you know very well. So this R is fused with this R will forms the, first we are going to take gametes in the Punnett square. That is very important. So then these two plants are used in the Punnett square. So now I will draw the Punnett square. See, this is the Punnett square. I will take gametes. Already know what kind of gametes it produce. Capital R one gamete. Here small r one gamete. Here also capital R one gamete and small r one gamete. So these gametes are taken into the Punnett square. First I will take these two gametes. I will write on the top of the Punnett square. Capital R and small r. Same the second type of gametes I will write on the left hand side of the Punnett square. So capital R, small r. Okay. When we fuse the gametes to each other, capital R is fused with the capital R. What kind of gametes we are seeing here? Capital R, capital R. Then capital R is fused with the small r, capital R, small r. Then small r is fused with capital R, small r. Then small r is fused with the small r. What kind of gametes? Small r, small r. So these four plants are F2 generation plants. These four plants are F2 generation plants in incomplete dominance. Remember, incomplete dominance. Remember, it is not complete dominance, it is the incomplete dominance. Okay, find out the phenotypic ratio of the F2 generation in incomplete dominance. So phenotypic ratio. What is the phenotypic ratio? Phenotypic ratio. So what is the phenotypic ratio? Visible characters. So what kind of flowers we are seeing in the F2 generation? See, capital R and capital R indicates it is which kind of flower? It is a red color flower. Both are dominant. Then coming to here, the capital or small r indicates it is which type of flower? It is not red color. Why it is not red color? Already I explained here. It is a pink color flower. Incomplete dominance. This gene produces only 50% of the protein. So when it forms the red color, it needs the 100 grams of the protein. But here there is no 100% of protein. For that reason, 50% it forms the intermediate character. What is the intermediate character? This is a pink color flower. So same like that, capital R small r indicates pink color. Here small r indicates white color flower. This is called what? White color flower. Remember. Then phenotypically, what is the ratio? See what type of plants. So how many red color flowers? 1. How many pink color flowers? 2. How many white color flowers? 1. So what about phenotypic ratio? In F2 generation, 1 is to 2 is to 1. Remember, compulsory in the incomplete dominance, in the F2 generation, what is the phenotypic ratio of F2 generation means? 1 is to 2 is to 1. So coming to the genotypic ratio. Genotypic ratio. So what is the genotypic? That means genetic makeup. See, same kind of genes, capital R present in where? Only one plant only. So I will write here 1 is to, see, this is only one type of plant. This is only one type of plant, and also see these two are similar genes, capital R smaller. Here also capital R is smaller. So this is called another type of genes. So how many plants same kind of genes? 2 plants. See, these two plants having same genes. Then this one plant having same genes. This one plant having same genes. So what is the genotypic ratio of the F2 generation in incomplete dominance? 1 is to 2 is to 1. Remember, here also 1 is to 2 is to 1. In the incomplete dominance, the phenotypic ratio and genotypic ratio of the F2 plants both are similar. Remember. Now I am going to discuss about another type of experiment that is called codominance. Okay students, first you will take screenshot. Okay, now coming to the codominance. See, codominance. Codominance. See, what is codominance? Codominance, it is in this process both alleles are expressed here. So that means both alleles are expressed. That means not only dominant character, not only recessive character. So both are expressed here. This codominance is a exception to the incomplete dominance. Remember, it is a exception to incomplete dominance. Remember, it is exception to incomplete. Incomplete means what? The intermediate characters we will see in the next generation. But here not like that. It is exception to incomplete dominance. So this kind of codominance we are seen in the human blood groups, where we see these human blood groups. Human blood groups. So already you know very well how many types of blood groups are present. There are 4 types of blood groups. What are they? First one, blood group A. Blood group A. And second one is blood group B. And third one is blood group AB. Blood group AB. And fourth one is blood group O. Remember, human blood consists of, all humans, they consist of 4 different types of groups. What are they? Blood group A, blood group B, blood group AB and blood group O. Okay, how to determine, how to detect the blood groups and what kind of arrangements we are seen in the blood groups. See, for example, this blood group A, in the what kind of alleles are present in the blood group. See, for suppose I will take chromosomes here. So these are the chromosomes, and inside of the chromosomes consists of double stranded DNA. Here also double stranded DNA, and it consists of alleles. For example, if you take blood group A, so which type of allele present? The alleles are present in the same location in the 2 chromosomes. For suppose I will take this location of the chromosome. So this location of the chromosome or DNA, it consists of which type of allele? Means A allele. Both are consist of AA. So here also allele A, here also allele A. This kind of chromosomes are seen in the blood group A. Or some other type is also present. So what are the other type of? Means see, some of the chromosomes, they are having, these are the chromosomes, and inside of the chromosome consists of double stranded DNA, and also the DNA consists of genes, and the genes consist of allele. So here which type of allele is present here means here one chromosome having A allele and second chromosome having I allele. So these types of alleles we are seen in the blood group A. See, always A is dominant than I. I means which type of allele it is means it is a defective allele. It is not expressed. Only here all A are present in the blood groups. So all A's are all A. It belongs to the blood group A. So what kind of antigens are present in RBC? See, for example, I will take the RBC cell. So this is the RBC cell. The surface area of the RBC cell, it consists of the some kind of proteins. What are the proteins means glycoproteins like this. These and all, the surface area of the RBC consists of proteins. So these proteins consist of antigens. Which type of antigens? All antigens are A. All antigens are A. Remember. So what kind of antigens are present in the RBC of blood group A? All type of antigens are A. So these are all antigen A. So this is what antigen A. So this kind of blood group we are called as a blood group A. Okay, coming to the second type of blood group. I will take what kind of chromosomes or allele are present here. See, these are the chromosomes, and inside of the chromosomes consist of double-stranded DNA, and in the blood group B consists of both are B allele. See, here also a part of the allele, a part of the gene. Here also a part of gene. So this part of the gene consists of B allele. Here also B allele. Or sometimes what kind of alleles are also present here means, see here these are the chromosomes, and chromosomes, inside the chromosome consists of double stranded DNA, and portion of the DNA we are called as a gene, and this gene consists of two different types of allele. What are they? One is B allele. This gene consists of B allele, and second one I allele. So the chromosomes which consist of B allele, two B alleles are B I allele. So this kind of blood group we are called as a blood group B. Then what kind of antigens are present on the surface area of the RBC? So this is the RBC, and the surface area of the RBC, it consists of the all are proteins. Already you know these are all proteins. So which kind? These proteins consist of some antigens. What kind of antigens? All are B. Here also B, here also B, here also B. So this is antigen B. This is called what? Antigen B. Okay, coming to the blood group AB. When we observe the chromosomes in the blood group AB. So in the blood group AB, what kind of alleles are present means here both are that means allele A present, allele B present, both are present. See, inside of the chromosome consists of double stranded DNA. A part of DNA we are called as a gene, and this gene consists of the alleles, part of DNA gene. So which kind of alleles are present here? Both A and B. Here not only A is dominant, not only B is dominant, both are dominant. For that reason, this kind of blood group, it gives the codominance. Codominance. Remember. So this indicates the codominance. So what kind of antigens are present on the RBC? So this is the RBC cell. Above the RBC cell having proteins, and the proteins consist of two types of antigens. Antigen A present, antigen B present. Again antigen A, again antigen B. So here both antigen and both antigen A comma B present. A comma B present. So both are dominant here. Not only single is dominant, both are dominant. So both antigen and both antigen A and both antigen B both are present. So this is about the codominance. Then coming to the blood group O. So when we observe the chromosomes in the blood group O. So these are the chromosomes, and inside of the chromosomes consist of double stranded DNA, and also a part of DNA we are called as a gene, and this gene consists of here there is no alleles in this blood group O. There is no alleles. So that means here what kind of, say for example, here or which type of alleles are present here means these are the chromosomes, and inside the chromosome consists of double stranded DNA. So both alleles are I. Remember, both alleles are I. There is no A allele, there is no B allele. Remember that. Both alleles are I. So when we observe the RBC cell, see here also proteins are present, and also here absence of antigens here. Absence of antigens. So absence of antigens. Okay, see, this is about the codominance when we observe in the human blood group. So the remaining concept I will discuss in the next class. Okay students, if you like my video, please subscribe my video, share this video to your friends and relatives. Okay, thank you very much.